Today we'll be talking about vision, electromagnetic energy. This is energy. There's an energy spectrum that includes X-rays, radar, and radio waves. There's a very small portion of this spectrum that includes light visible to the human eye. So light can be described both as a particle and a wave. So we think about light as just, whatever we can see. But as I'll show you on the next slide, what we can see is actually a very small part of it. I'm sure you guys have heard of like X-rays and radar and infrared rays and all these different things. Well, we can't see that, right? So what we can see is actually a very small part. So here's just an example of it. So you see this whole thing, you got gamma rays, X-rays, ultraviolet rays, and then you see the visible light, right? You see the electromagnetic spectrum that we can actually see, this small little part right here. Then you got infrared rays, radar, broadcast bands, AC circuits, things like that, which is amazing that, we actually see a very, very small part of that. Light energy, right? So light comes in waves. Two vocab words to know: wavelength. Make sure you know that's the distance between peaks that determine hue. All right, so the distance between these wavelengths right here, that determines what the hue is. Remember, hue is just whatever color you experience. Amplitude is the height of peaks determines the intensity of the color. So how high these peaks go determines how intense those colors are. So the distance is the wavelength. That's what color it is. But there's like light and dark blues and reds and things like that. That's determined by the amplitude. Just remember, the human eye can see over 7 million different types of hues. So a lot more than that crayon box you had as a kid. Hue. Like I said earlier, hue is the color of light as determined by wavelength of the light energy. So this includes, red, orange, yellow, green, blue, indigo, and violet. I'm sure you guys have probably heard of Roy G. Biv. All right, that's one way to remember the 7 main colors. Like I said a second ago, the eye can detect 7 million separate hues. If you look at the picture there, notice the short wavelength. So we talk about wavelengths determines hues. Well, a shorter wavelength equals higher frequency when we talk about hearing. But when we talk about color and what we see, those are bluish colors. Right? So high-pitched sounds, high frequencies, and bluish colors for short wavelengths. Longer wavelengths equal low frequency, so low-pitched sounds, and reddish colors, and obviously in everything in between. Vision. So what's the purpose of the visual system? Right? Transduction, vocab word, very important. So this is transforming light energy into an electrochemical neural response, right? That's action potential, that's synaptic transmission. So remember, your brain can't read waves. So sound waves, visual waves, that's how they're processed, but your brain can't read that. So it needs to be transformed into an electrical impulse that your brain can actually read. And that's called transduction. So remember, happens in your eye, we're going to talk about in a second, that's in the retina. And remember, in the ear, it happens in your cochlea. Two important things to know. And so the purpose of the visual system, like we said, is to represent characteristics of objects in our environment, such as size, color, shape, and location. But you can't do that without transduction, right? If in the eye, if we're not changing that wave into an electrical impulse that your brain can understand, then we wouldn't be able to see. Focusing on light. So here's kind of the anatomy of the eye. So it's kind of important to know the different parts of the eye. So the first part is the cornea, the protective covering. So that's the outside part of the eye. So you might also have to know kind of like the process of how light goes through your eye. Well, that's going to be the first part because that's the outside part. So that's the protective part. If you've ever scratched your eye, that's your cornea that got scratched. Your pupil. This is the opening in your iris where light enters, allowing more or less light to enter your eye. So if you're in a dark room, your pupil gets bigger to lend in more light. If you're in like outside and it's bright, your pupil is going to get smaller because not to lend in not as much light because it's so bright. So once again, your pupil changes, right? It's inside your iris. Remember, the iris is the colored part of your eye. And so it gets bigger in dark areas and smaller in light areas. The lens bends light onto the retina, focuses the image on the back of the eye. All right, so if you don't have 20/20 vision, that's the lens is bending the light onto the retina, it's not focusing it properly. Whether it's too soon or too late, we'll talk about. But that's why you might need glasses. And or if you have like LASIK eye surgery, eye correction surgery, like I've had a few years ago, that's what they do. They change the shape of it so it bends it correctly. The retina, you see there in the back of the eye, contains receptor cells sensitive to light. Retina is very important. We'll talk about in a second. The optic nerve exits eye to take information to the brain. So you see that in the back of the brain here. All right, so that's where it exits the eye. We're going to talk about that also shortly because you have something called a blind spot, and that's where your blind spot is located, and I'll discuss that in a second. So how can you remember all these? Like I said, if you can create an acronym, create your own acronym. You're going to remember it better. I find these stupid ones. If it helps you, it helps you. You'll also notice as we go through some of the slides, you're going to see these acronyms on the slides. That's why. So how can you remember the parts of the eye? Can people itch little rodents on necks? All right, so you got cornea, pupil, iris, lens, retina, optic nerve. So as we go through, like here, you see we're talking about the retina, you see rodents in parentheses because that's the part of the acronym to remember. But remember, if you can make up your own, I mean, you're going to remember it better. All right, so the retina. Retina is extremely important. So what is it made up of? It's made up of cones and rods, bipolar cells, and ganglion cells. Make sure you know the difference between rods and cones. Cones is what allows you to see color. All right, so cones allows you to see color. Rods allow you to see light and dark. So animals that are mostly nocturnal, they mainly have rods in their eyes because that's what allows them to see in the dark. So humans can see in the dark because we have some rods, not as well as other animals though, but yet better than some animals can't see at all in the dark. We can see in color. Not all animals can see in color. We can see in color because of cones. If you've ever seen the Blue Man Group, they have a good little show that I'll post, and it kind of goes over the rods and cones, and you can see the layers there. Bipolar cells. These send messages to the ganglion cells from the cones. The ganglion cells then take the message to the optic nerve. And you can kind of see the layering, how you have it there. And notice how the cones only have certain colors, and we'll talk about that in a little bit as well. So this is very important because this is where transduction occurs. So you've heard me say transduction a lot this unit. It's extremely important when we talk about the eye and the ear because sound waves, visual waves cannot be read by the brain, so you must transduce them into an electrical impulse that the brain can understand. And so in the eye, it happens in the retina. All right, so we talked about transductions where light is absorbed in and transformed into these electrical chemical signals used by the nervous system. The Hermann grid. All right, so if you stare at it, just stare at the grids for a second. Do you see gray dots in between the squares? You should. Now, there aren't really gray dots there, but the rods in the peripheral are responsible for this. Because when you look at an area directly, there's no dot because you are using your cones. Remember, your cones is what allows you to see color. But the peripheral has dots because the rods are trying to do two things at once. Show you that there's a dark spot and a light spot. So when you just stare at it, there's black and there's white. Well, your rods try to tell you that it's light and dark, so that's why you get like these gray spots. I'm sure you've seen it, somewhere when you've stared at things before. And the reason why you get those gray spots that aren't really there is because your rods are trying to do two things at once and it can't, and so it kind of blends together and you get gray spots. But if you look directly in between the squares, obviously you just see white. That's because you're using your cones now. The fovea. The fovea is the central focal point of the retina. This is the spot where vision is best, right? The most detailed, something called visual acuity. We'll mention this again later as a little review. Only cones are found in the fovea. So this is like the central part of the retina. Only cones are there, and because of that, this is where your vision is best. In a vocab word, that's called visual acuity, where vision is best. Make sure you know that's the fovea. The optic nerve. All right, the optic nerve is the nerve that carries visual information from the eye to the thalamus and then on to the occipital lobes of the brain. So if you remember when we talked about the brain, the thalamus is your relay station. All your senses go through your thalamus except for, hopefully you said smell, and then your thalamus sends it to whatever part of the brain it needs to go to. Well, in this case, if you remember, the occipital lobe is in the back of your brain. That's the one that deals with vision. So if you ever get hit in the back of the head and it kind of goes black for a second or you see stars, that's because that's what deals with vision. So in this picture, you can see the retina, right? You see the fovea is the very middle part of the retina. This whole thing is the retina here. And you got the optic nerve, which takes it, leaves the eye, takes information to your brain. All right, so we've talked about the optic chism before. That's just the opposite. So if you remember when we talked about the brain, everything on the left, so your left brain controls everything on the right side of your body, including your right eye, and vice versa. So the chism is just where they cross, right? So everything coming from your right eye goes to your left brain, your left eye goes to your right brain to be processed. So, like I mentioned earlier, you actually have a blind spot. So where your optic nerve leaves the eye, there's no rods, no cones at this point. So you actually have a blind spot in your vision. You don't notice your blind spot because each eye compensates for the other, or your brain just kind of fills in the missing information. So you'll see this in the textbook. Usually I have a handout. Hopefully I can get you guys. So what you do is on the handout, there's a plus and a circle, and you stare at one. And as you move it closer or farther away from you, eventually the circle or the plus sign, the cross, will disappear. And wherever it disappears at, that is your blind spot, right? It's just going to look blank there unless you move your vision over to actually stare at it. But if you stare at like the circle on the one side, and your book will talk about this, and I can send this to you guys. It literally, you can see your blind spot. And that's because you have no cones or no rods in there, because that's your optic nerve. It's sending information to your brain. Now, you don't see a blind spot in your vision. That's because your brain, like we've talked about, is always trying to make sense of everything. So it literally just fills in background information. So if you're staring at a white piece of paper, it just looks white, right? So we don't have blind spot, or we do have blind spots, but we don't notice it because your brain is just filling in that information. So students love this. It freaks them out sometimes because no one realizes that you have a blind spot in your eye until you do this, and all of a sudden it like disappears on you. So I always have a couple students that want to bring it home and show their parents. So we talked about there's a pathway. So obviously there's a lot here, and hopefully you can remember as much as possible, but make sure you remember the big parts of it. So remember the cornea we talked about is the outside part of your eye, then it goes to the pupil, then to the lens, then to the fovea, which is in the retina, then to the rods and cones, then to the bipolar cells, then to the ganglion cells, which have movement and light, color and detail. Then that goes to the optic nerve, that's where your blind spot is. Then to the optic chism, remember that's a crossover point. Then to your thalamus, because your thalamus processes all of your senses except for smell. And then it goes to your occipital lobe, right, because that's your primary visual cortex. Now, how can you possibly remember all of that? So we talked about the acronym earlier. Here's another one for you. Once again, I know they're corny. They're just whatever I found. Once again, please create your own. If you create your own, you'll take more ownership of it and you'll remember it. And I've had some students create some cool ones in the past. So here's one to remember all of that. Cool people like Frosties because gangsters never cheat the officers. Right? So you got cornea, pupil, lens, fovea with the rods and the cones, bipolar cells, ganglion cells, then the optic nerve, then the optic chism, then the thalamus, and then the occipital lobe. Once again, that's just an example for you. Please choose your own to remember this.